Hello and welcome to the second presentation of our All Hands EPSCoR meeting. I'm Sam Richardson from Dr. Shao's group.
And I'm Denver Landers from Dr. Jarbo's group. It's great to have you all here today. Now let's get started with our topic for the day. What are metabolic engineering and synthetic biology, and how are they used in biomanufacturing?
Throughout human history, we've always relied upon other organisms to survive. All of our crops and livestock have been domesticated from some sort of wild progenitor. We take this progenitor and over generations of selective breeding, shape the organism for our needs. Whether it be sheep's wool for clothes or yeast for beer, humans have always altered the genetic destiny of organisms we find useful. Synthetic biology and metabolic engineering allow pathways for the production of desired materials, improving existing ones or creating new ones, all together with great precision. An example of this domestication is maize, better known as corn, from its wild progenitor teosinte. The ancient corn breeders practiced selective breeding techniques by saving seed from plants that had desired traits and replanting those seeds for the next harvest season, eventually leading to the large, delicious corn cobs of today. This was accomplished over hundreds of years. With synthetic biology and metabolic engineering, this could be achieved in a significantly shorter time frame with directed alteration of desired traits, cutting down on the randomness of genetic change. The goal of synthetic biology and metabolic engineering is to produce the microbial factories necessary for biomanufacturing. Biomanufacturing involves the use of biological systems that have been engineered or used outside their natural context to produce a product. Biomanufacturing is one of the most important methods of production today, as it has the potential to utilize sustainable biomass, such as corn, from across the United States for conversion into new products, and provides an alternative to petroleum-based production for fuels such as ethanol, commodity chemicals like synthetic rubber, and green materials, medicines, and more. The cellular material used for this manufacturing may be unmodified, but is usually a result of focused metabolic engineering to shape an organism into an easy and efficient producer of a needed product.
Metabolic engineering is similar to synthetic biology because it uses genetic manipulation to affect the productivity of biosynthetic pathways, but its motivation is more outcome driven rather than seeking understanding. In metabolic engineering, the manipulations performed on the genome of the host organism are designed to increase the productivity of the desired product and to reduce its consumption in accessory biosynthetic pathways. Metabolic engineering uses manipulation of the culture environment in addition to genetic manipulation to attain its goals, and it can do this through equipment design. There are four broad categories of metabolic engineering: to enable new behavior, eliminate undesired behaviors, tune enzymatic activity, and lastly, to generate diversity that may produce desirable properties. This last category of metabolic engineering acts as a shotgun approach to altering an organism. It allows many different versions of that organism to be sifted through in order to find useful variants.
Examples of the four broad categories of metabolic engineering include insulin and yeast. We can insert genes that make an organism do something new. For example, produce insulin. Before 1978, insulin from cows and pigs was used, but was hard to collect and caused harmful allergic reactions to humans. By replacing parts of E. coli's genetic code with the human gene that allows the production of insulin, synthetic human insulin could be produced for the mass market, saving many lives. We can also insert genes to eliminate undesired production or utilization of a compound, such as altering yeast to only feed off of certain sugars, or reduce enzymes that produce alcohol in fermentation to prevent ethanol-induced yeast death.
To discuss the toolkits available today for engineering and culturing biological systems, we first have to speak to what is called the central dogma of biology. The central dogma of biology is the premise that genetic information for the development, regulation, and maintenance of the organism is stored in DNA. DNA is copied through the process of replication. DNA is read and used for cellular processes through the process of transcription, in which RNAs are made from the template DNA. These RNAs are used in many different ways by an organism, but one of the more direct ways they are used is in the production of proteins. So-called messenger RNAs are read by ribosomes and translated into proteins. Proteins have many uses, but the two categories they primarily fall into are structural and catalytic, which are called enzymes. Enzymes are the machinery that are employed to transform precursors into intermediates, and eventually into products, which are our desired chemicals. When multiple enzymes are used to transform a material like glucose into some product, this is called a biosynthetic pathway. With all of this complexity, we need analytical tools to determine how resources are being used by a cell, and this is where the omics come in. The broad set of omics are ways of measuring the resources within a cell at different time points, and these different time points can be used to understand how cells develop and respond to changes in their environments. The genome is a collection of all genetic information in the organism. The transcriptome is the complete collection of RNAs in a cell at a given time. The proteome is a collection of proteins in a cell at a given time, and the metabolome is a collection of the small molecule chemicals called metabolites present in the cell at a given time. Metabolites include chemicals like amino acids, nucleic acids, fatty acids, sugars, vitamins, and cofactors. Synthetic biology is a field of study that manipulates the genetic code of organisms with the understanding of the central dogma of biology. And through this, they seek to better understand how biological systems work at large. It often investigates different genetic elements from a systems approach. Where we often think of genes as discrete units, they're actually composed of multiple elements. Those are promoters, encoding, and terminator regions. Synthetic biology concerns itself with the study of these structures, as well as the overall organization of genes in the chromosomes, and the knowledge it acquires in the process is then shared with other scientific disciplines. It accomplishes this work with a method similar to the design, build, learn, test method of other engineering disciplines.
While synthetic biology and metabolic engineering are similar, there are distinctions between the two that are important to be aware of. Demonstrated by this Venn diagram, while both approach problems from an engineering mindset, the scope of each field is drastically different. Metabolic engineering has a focus on finding useful pathways within organisms that we can exploit to create cell factories in order to achieve some desired product or behavior. Much of this is done by changing gene expression using the toolkits mentioned prior. Synthetic biology has a much broader scope, with the focus being on ground-up construction of new pathways, with a fantastic recent example being in the Impossible Burger. As any of us who enjoy meat know, blood greatly influences flavor, and is obviously very hard to get out of a plant-based patty. Impossible Foods engineered their yeast by moving a large cellular pathway from soy that produces the legume version of hemoglobin, leg hemoglobin, providing a better meat taste and eating experience.
As Sam said before, we have shaped organisms throughout history through selective breeding. Today, we still use selective breeding under its own name and others like directed evolution, but now we have additional tools at our disposal. Naturally, with time, organisms acquire mutations in their genomes as a result of environmental stresses like radiation and chemical interactions. These random mutations can be passed on through reproduction. This is the crux of selective breeding and has been the paradigm of organismal manipulation for millennia. Recently, the CRISPR-Cas9 system and other endonuclease systems have been discovered, all of which cut DNA at specific locations determined by their natures. This allows for extremely precise gene editing, and all of this allows for a more intentional design approach. With these systems, we are able to completely delete the effect of whole genes, which are generally referred to as knockouts. We are also able to substitute partial regions of encoding regions of genes with alternative sequences to change the amino acid sequence of the resulting proteins. And we can substitute whole promoter regions, which can cause more or less transcription, all of which affect the metabolism of the organism. Combining these more precise technologies with selective breeding results in our ability to vastly improve the metabolism of organisms for a desired purpose. These could be as simple as improved drought tolerance in crops, or producing plastic ingredients and microbes.
With all of that, we would like to thank you for your time, and we hope that you have learned something about synthetic biology and metabolic engineering.